So, have you ever really stopped to think about what holds you together? And I don't mean your skeleton. I mean way, way down on a microscopic level. Our body's tissues aren't just these mushy bags of cells. They're actually more like a bustling, incredibly organized city, complete with skyscrapers, superhighways, and complex communication grids. In this explainer, we're going to pull back the curtain on the incredible architecture that makes all of that possible.
And that brings us to the big question, right? What is it that gives our tissues their specific shape, their strength, and allows them to do their job? How does a liver cell, even though it's in a liver, or a skin cell know it's part of this flexible protective barrier?
Well, the answer is hiding in plain sight. It's this amazing universe of scaffolding. And it's really a two-part system. There's an internal, living skeleton inside every single one of our cells, and then there's a complex, totally functional matrix on the outside that kind of glues everything together. You put those two together, and you've got the fundamental infrastructure of life.
Okay, so here's our game plan. We're going to start by looking at the cell's internal skeleton. Then we'll see how that very same skeleton acts as a highway system. After that, we're going to step outside the cell into the matrix. Then we'll see how the inside and the outside are connected. And finally, we'll get into the really critical stuff. What happens when this whole scaffolding system fails?
All right, first up, the cell's living skeleton. Now, this is not some static, bony structure. It's a super dynamic network of protein filaments we call the cytoskeleton. And it gives the cell its shape, its strength, and its ability to move around. And this table here gives us a fantastic look at the three main players on this team. First, you've got microfilaments. These are the thinnest ones, made of a protein called actin, and they're absolutely essential for things like cell crawling and muscle contraction. Then, in the middle, we have the intermediate filaments. Think of these as the steel cables of the cell, providing just raw tensile strength. And, super important, they're unique to different cell types, which, as you'll see, is a huge deal for diagnostics. And finally, the big ones, microtubules. These are the structural girders, but they also act as the cell's main highways for transport. We'll get back to that in a second. So how does the skeleton let a cell move and change its shape? Well, it uses two really clever strategies. On one side, you have actin polymerization. Just picture the cell laying down new bricks right at its leading edge, literally pushing the membrane forward. That's how a cell crawls. On the other side, you've got microtubule dynamics. These things are constantly growing and then shrinking back down. It's a process called dynamic instability. This lets them basically explore the inside of the cell, grab onto chromosomes during cell division, and pull them apart. It's this brilliant system of pushing versus organizing and pulling. Now let's build on that. Those microtubule girders we just talked about, they aren't just for structure. They form the cell's entire logistics network, a vast system of highways for moving all sorts of cargo around. And every highway needs vehicles, right? Well, the cell has them, molecular motors. These are incredible little proteins that do exactly what it sounds like they do. They take chemical energy from ATP and turn it into mechanical force. They literally walk, step by step, along those microtubule tracks, carrying little packages of cargo to wherever they need to go. Let's break down how one of these motors, kinesin, actually walks. Think of it having 2 feet. One foot grabs onto the microtubule highway by binding to an energy molecule ATP. That locks it down tight and causes the other foot to swing forward. Then, that first foot uses up its energy, lets go, and swings around to the front to take the next step. It's just this constant energy-burning cycle of grab, swing, release, grab, swing, release. And this is where it gets really sophisticated. It's not a random walk, it's a fully directed GPS. There are 2 main families of these motors. Kinesin generally walks outward, from the center of the cell toward the edges. Dyning does the opposite. It walks inward, from the edges back toward the center. Now, from a clinical standpoint, this is a major vulnerability. Viruses like rabies and herpes are absolute masters at hijacking the dyning motors to catch a ride from a nerve ending all the way back to the main cell body where they can set up shop and replicate. Pretty clever, huh? Okay, so we've covered the cell's internal architecture. Now, let's step outside, into the world that surrounds it, the extracellular matrix, or ECM. And let me be clear, this is not just empty space. It is a functional, dynamic, and absolutely vital environment. The ECM has a bunch of critical jobs. Of course, it provides mechanical support, giving cells something to anchor to, but crucially,
It also controls when cells grow by acting like a storage depot for growth factors, holding onto them until they're needed. It provides the essential blueprint for tissue repair after an injury. In fact, if the ECM gets destroyed, organized repair becomes basically impossible. And finally, it creates specialized little neighborhoods, like the ultrafine filtration barrier in your kidneys. Now, the ECM basically comes in two main flavors. There's the interstitial matrix. This is the stuff you find in the spaces between cells in connective tissue. Think of it like a squishy 3D gel. Then by contrast, you have the basement membrane. This is a highly organized, super thin, flat mat. It literally looks like a sheet of chicken wire that sits right underneath layers of cells, providing this critical boundary and support structure for them to rebuild on. So what is this matrix actually made of? Well, it rests on three main pillars. First, you have collagen. It's the most abundant protein in the human body, and it forms these rope-like fibers that provide incredible tensile strength. They're amazing at resisting being pulled apart. Then there's elastin, which is exactly what it sounds like. It lets tissues like your skin and your blood vessels stretch and then snap right back. And finally, you have proteoglycans. These things are wild. They form these super hydrated gels, almost like a sponge, that give tissues the ability to resist being squished. So we've seen the inside of the cell and we've seen the outside, but here's maybe the most important part. How do they talk to each other? How does a cell actually connect to and sense the world around it? The link is made by a family of proteins called integrins. And just like their name says, they integrate the inside world with the outside world. They sit right in the cell membrane and physically bridge that gap, linking the internal actin skeleton to proteins in the external matrix. This is how a cell literally feels its environment. When integrins on the outside grab onto the matrix, they all cluster together on the inside. This creates a structure called a focal adhesion. This complex then latches onto the actin cytoskeleton, making a really strong anchor. But here is the absolute magic. This physical connection isn't just mechanical. It translates that external physical tug into a cascade of chemical signals inside the cell by turning on signaling proteins. This is how the physical world of the matrix directly tells the cell what to do. And now we can really start to understand disease, because so many pathologies are at their very core just failures of this cellular architecture. Let's take a look at what happens when the scaffolding starts to crumble. A perfect and devastating example is fibrosis. You see, in chronic inflammation, certain signaling molecules just go haywire. They hijack the normal repair process and basically scream at the cells to deposit way, way too much collagen. The result is scarring, fibrosis, and it destroys the normal architecture and function of our most vital organs, like the liver, lungs, and kidneys. And this slide just drives the point home with some powerful examples. A tiny mutation in lamin, one of those intermediate filaments that supports the nucleus, can cause horrific diseases like progeria, that's premature aging. A defect in type 1 collagen, you get brittle bone disease. If your dining motors don't work, your cilia can't move, leading to Cargener syndrome. And on the flip side, for diagnostics, since we know intermediate filaments like keratin are specific to certain cells, we can use them to figure out where a cancer came from. And the list of these architectural failures just goes on. A defect in a protein called fibrillin, which helps organize elastin, leads to a weakened aorta in Marfan syndrome. Something as simple as a vitamin C deficiency is a disaster because you can't make collagen properly. That leads to the weak blood vessels and terrible wound healing you see in scurvy. And as we mentioned, the dysregulation of that signaling molecule TGF-beta is a central bad guy in fibrosis across tons of different organs. So when you boil it all down, the takeaway is this. From the shape of one single cell all the way up to the function of an entire organ, our health is fundamentally built on this incredibly intricate and dynamic architecture. Understanding it is absolutely key to understanding disease, and hopefully to one day designing better ways to treat it. Which really leaves us with one final fascinating thought. We've seen how the ECM provides the critical scaffold for tissue to renew itself. So, as our understanding of this cellular blueprint gets deeper and deeper, it begs the question, can we one day move beyond just being repairmen and become the true architects of our own tissues?